Hello and welcome everyone who's joining our masterclass today. I am Dr. Lea Esser and I am part of the research and development team here at Cube Biotech, responsible for protein purification and activity assays. And today I'm honored to host the Intelligent Infusion Masterclass from Cube Biotech, which was created for researchers to give them the opportunity and a platform to share and promote their research with a worldwide audience. After the talk, just some organizational stuff, there will be time for an interactive question and answer section. You can therefore type in your questions during the talk in the lower right part of the interface with the question mark, and afterwards they will be shown on the screen. And today, with this, I'm happy to introduce our speaker, Professor Dr. Sara Garcia Linares. She earned her PhD in Biochemistry and Molecular Biology from the Complutense University of Madrid in 2017. She completed a 2-year postdoctoral stay at Harvard Medical School, where she structurally and functionally characterized one of the proteins involved in the Hedgehog signaling pathway. She is currently a professor and researcher in the Department of Biochemistry and Molecular Biology at the Complutense University of Madrid. Her research is focused on studying the structure-function relationship of toxic proteins from sea anemones and insect cells, with a particular interest in their interaction with biological membranes and the role lipids play in these interactions. She is also a member of the board of directors of the Spanish Society for Biochemistry and Molecular Biology. She has a strong passion for science communication, and she has also been involved in and led educational innovation projects. She has combined her scientific career with performing in various orchestras as a violinist. And now I am happy to hand over to Professor Dr. Sara Garcia Linares, who hear her interesting research talk with the title Using Nanodiscs and Functional Characterization of Toxic Proteins. And with this, I want to hand over to Sara.
Thank you, Leah, for the kind presentation. Good afternoon, everyone, and thanks to Cube Biotech for inviting me to give this masterclass. Let me change my pointer so everyone can see it. Okay, so in the next few minutes, I will explain how we have harnessed the use of nanodiscs for the structural and functional characterization of toxic proteins. Moreover, we have used these nanodiscs to transform these pore-forming toxins into powerful biotechnological tools. So our final goal is to develop a sustainable technology to remove and recycle plastic particles, both in wastewater and drinking water, equipping water treatment plants with innovative filtering devices so we can remove micro and nanoplastic particles. We all know that plastic pollution is a pressing problem in our society globally. Each year, around 270 million tons of plastic are produced, and due to improper management of residues, around 8 million tons end up directly dumped into the oceans, which means that we have around 10 to 100,000 tons of plastic in the surface of our seas and oceans. And this problem is even worse because over time, these plastics degrade into smaller fragments that we know as micro and nanoplastics. And these tiny particles have completely invaded our planet. They are in the air, they are in the water, they are in remote areas as Antarctica. And we can even find them in the food we eat and the water we drink, because as these plastic fragments break down into ever smaller particles, they can be ingested by different organisms, and that makes it easier to find their way all to the top of the food chain. So one of these plastics is PET, which is the polyethylene terephthalate, So to put it simply, is the plastic that we can commonly find in transparent plastic containers or bottles. And this material accounts for approximately 10% of the total plastic production in the world, and its recycling is still very inefficient. So in this seminar, I'm going to tell you how we have harnessed the potential of nanodiscs, here in gray, as lipid platforms to put a protein nanoreactor, in gold, capable of hydrolyzing PET nanoparticles, here in blue. And what is this protein nanoreactor? So it comes from the venom of a sea anemone. These animals have a specialized type of cell called cnidocyte. And inside the cnidocytes, we can find nematocyst, which is an organelle that contains the venom of the sea anemone. So whenever a prey comes near enough the sea anemone's tentacles as to touch this trigger here called cnidocyl, then the nematocyst would eject from the cell and shoot out, like discharging a harpoon. So there's a funny animation by TED Education that we are going to watch that perfectly illustrates this process.
When mechanical or chemical stimuli activate an external trigger, the lid of the cell pops open and seawater rushes in. This forces a microscopic barbed harpoon to shoot out, penetrate, and inject venom into its victim.
So if you ever touched a sea anemone or a jellyfish, you know that that's really painful. And that's because the venom of the sea anemone or the jellyfish is composed of a complex mixture of toxic small proteins. We can find mainly neurotoxins, which are toxic to neurons, and they are responsible for the pain that we feel when we touch one of these animals. But we can also find phospholipases that degrade the lipid bilayer of our membranes. And finally, we can find cytolysins, which are proteins that make holes in the cells, and they are responsible for the inflammation of our skin when we touch one of these animals. So in this last group of toxic polypeptides, we can find a family of proteins called actinoporins. Actinoporins are proteins composed of a single polypeptide chain of around 175 amino acids. They are pretty small. They don't have cysteine residues, and they have a basic isoelectric point. And all of them share an almost identical three-dimensional structure consisting of a beta sandwich flanked by two alpha helices. So far, actinoporins have been isolated from at least 20 different sea anemone species, but only 4 of them have been thoroughly studied, and those are echinatoxin 2 from Actinia echina, sticolysins 1 and 2, both from Stichodactyla elianthus, and fragacetoxin C from Actinia fragacea. The mechanism by which these proteins make pores in the cells is still a matter of study, but there are some key processes that are commonly accepted. Mainly, actinoporins remain stably folded as monomers when they are in aqueous solution. But if they encounter a lipid membrane of the right lipid composition, they can bind to it. And when I say the right composition in the context of actinoporins, I mean the membrane must contain sphingomyelin. This lipid acts as a kind of a receptor for these proteins. So once they bind to the membrane, the N-terminal helix extends parallel to the membrane, and then it inserts into the membrane, while several monomers of the protein oligomerize to form the final pore, which is cation selective and creates an ionic imbalance in the cell that ultimately kills it by osmotic shock. So back in 2015, the atomic structure of the pore made by fragacetoxin C from Actinia fragacea was solved. And this pore, as you can see, is composed of 8 protein monomers, and the pore walls are made by the 8 N-terminal alpha helices of the proteins, but also of the membrane lipids that appear between the helices. And this structure was the most precise and accepted that we had for actinoporins, but it had a few drawbacks because this structure is a crystal structure obtained in the presence of lipid mimic detergents. So it doesn't really illustrate the real situation of the membrane pore process. And because of that, it didn't give much information about protein-lipid interactions. So that is why our research group turned to Nanodiscs to start investigating the structure of these pores in solution. We are all familiar with these particles. They are lipid platforms of nanometric size composed by a lipid bilayer that is stabilized by either a membrane scaffold protein or an artificial polymer. In our case, we use MSP-based nanodiscs, and to make them, we just mix the lipids dissolved in detergent with the membrane scaffold protein, and then we remove the detergent by dialysis. During detergent removal, the nanodiscs are formed, and the result is a particle that is completely soluble. Nanodiscs offer other advantages like uniform size, controlled lipid composition. You can use different methods of formation. We use dialysis, but you can use also size exclusion chromatography or biobeads. And the most important for us, there are no detergents in the final sample. So what we do is we use empty purified nanodiscs, and then we add actinoporins to reconstitute the membrane pore. So in our case, we do reproduce exactly how the biological process takes place. No detergents are involved, and the final pore structure is perfectly soluble. Actually, in collaboration with Jaime Martín Benito and Ernesto Arias, we got amazing images of cryo-electron microscopy of this membrane pore made into nanodiscs of both sticolysine 2 and fragacia C. So you can see the 8 monomers of the pore, of the membrane pore, and in this side view, you can even distinguish the secondary elements, the secondary structure elements, including the N-terminal alpha helices of the protein that will make the pore walls. So after a million of images were processed, we got a 3-dimensional structure for the pore with atomic resolution. And this structure has allowed us to determine atomic interactions between protein amino acids and the membrane lipids. The resolution obtained is one of the highest that has been achieved with membrane protein, and has allowed us to perform analysis of protein-lipid interactions to the point of distinguishing cholesterol and sphingomyelin individual molecules in the pore structure, highlighting their important role in pore formation. Moreover, we have captured, using nanodiscs, intermediates of pore formation, which have allowed us to postulate a mechanism of pore formation in this case. So how did we get from a protein of an anemone that forms tiny pores to biological nanoparticles to filter and degrade plastic particles? So Professor Guallar's team from the Barcelona Supercomputing Center have developed a software called Pele, Protein Energy Landscape Exploration, which integrates protein structure prediction algorithms with stochastic approaches to explore energy landscapes and map millisecond scale processes. So it's a technique very similar to conventional molecular dynamics simulation, but with much lower computational cost. So I'm going to give a very brief overview of how Pele works. So the whole process begins with a perturbation of the system. All of this is computational. So you put your selected ligand in the presence of your protein of interest. And then there's a relaxation process where a side chain prediction and a global minimization occur. That means that Pele identifies which amino acids of the protein need to be modified in order to interact with the ligand that you selected. And in the final step, the energy values of the entire process are evaluated to determine whether the proposed amino acid changes should be validated or discarded. So we selected 2 of the mutants that the software predicted using the structure of fragacia C in order to introduce specific mutations to introduce a new catalytic triad with enzymatic activity capable of hydrolyzing PET nanoparticles. And after the selection of the mutants, our research group produced the proteins using an E. coli system and a very optimized purification protocol for actinoporins, obtaining a good yield of either wild-type fragacetoxin C or the 2 mutants. So now we need to reconstitute the pore into the proper lipid platform, and then we turn again to nanodiscs. For this experiment, we use a complex lipid mixture composed of DOPC, sphingomyelin, which is the actinoporin receptor, and cholesterol, which is a non-enhancer for pore formation. So after purifying the empty nanodiscs, we added the actinoporin to reconstitute the pore. And by negative staining electron microscopy, you can see here an image where you can distinguish clearly a pore made into 2 nanodiscs, although the whole sample is full of them. So we have our nanoreactor ready to use. So for a preliminary test, we incubated agar plates with PET nanoparticles in the presence of either the wild-type fragacetoxin C pore as a control or the 2 mutants. And this was the astonishing result. You can see how the wild-type nanopore barely shows any activity against the nanoparticles, while both mutants display a clear hydrolytic effect on the nanoparticles. So after this amazing result, these activity tests were done at the Institute of Catalysis in Madrid. And for a more thorough analysis, they monitored the degradation of PET nanoparticles by HPLC to determine the concentration of the degradation products of the PET nanoparticles. So now we are going to watch a video where you can observe how the PET nanoparticle suspension becomes clearer as degradation progresses. Thank you. So both nanoreactors were tested with 4 different PET nanoparticle substrate, each with distinct physicochemical properties. The first mutant, located within the pore channel in the N-terminal helices that penetrate the membrane, it yields as a main degradation product BHAT. This is the main precursor of PET upcycling routes, which makes this mutant highly valuable for PET recycling. On the other hand, the main degradation product for the second mutant, located in the outer part of the pore, is terephthalic acid, which is the main constituent unit of the PET polymer, suggesting that this mutant could be useful for the final degradation of PET. Thus, we have 2 active mutants capable of degrading PET nanoparticles, each of them useful for different purposes, one of them for recycling and the other one for complete breakdown and elimination of PET. It is also worth mentioning that all of this was done at 40 degrees temperature and neutral pH, which offered a clear advantage over other existing PETases that work at 70 or 80 degrees, which increases CO production during the PET degradation. So let me finish with some important take-home messages. Nanodiscs are invaluable for studying membrane proteins, providing a controlled and native-like lipid environment. And in our case, the use of nanodiscs in the study of actinoporins has allowed us to reach 2 important conclusions. First, that lipid molecules are not passive participants, but active structural components of the pore made by actinoporins. And also that pore formation mechanism is governed by a specific lipid-protein and protein-protein interactions, offering potential for targeted biotechnological applications. We saw how actinoporins can be engineered for use in drug delivery, leveraging their specificity for particular lipid compositions. And as I showed you, they also hold promise for environmental applications, such as the enzymatic breakdown of pollutants like micro and nanoplastics. So that's what I wanted to share with you. And now I'll be happy to answer any questions you may have.
My colleague will be ready with the questions, so we're waiting for the first question to come. So here we have it already. So the first question is, what methods do you use to isolate and purify empty nanodiscs?
Yeah, after we have the mixture, after detergent removal, we use size exclusion chromatography We get a really big peak of lipid aggregates, and then a smaller one, but really, really clean peak for the purified empty nanodiscs.
Yeah, great. The next question is, how do the lipid composition and membrane properties influence the functional behavior and structural stability of actinoporins within nanodiscs?
Yeah, so lipid composition is critical for the activity and stability of actinoporins. We work with this complex mixture with sphingomyelin because we knew already that it's a receptor for actinoporins. That's been known for many years. But from our study with the atomic resolution structure, you can see sphingomyelin forming a stabilizing ring around the actinoporin, so it's essential to be there. And cholesterol also, it's important to have the proper membrane thickness and membrane physicochemical properties to enhance pore formation. So controlling the lipid composition and the environment is really important to have the proper pores formed for whatever use you want to make them.
Then the next question is, how does the mechanism of pore formation revealed by cryo-EM studies contribute to the potential biotechnology applications of actinoporins, such as in drug delivery or bioremediation?
So yeah, that's a really good question. For the pellet software, it's really important to have an atomic resolution structure that we could do that thanks to the 2015 structure. And now that we have the new one with all the information about the lipid-protein interactions, we better know which amino acids we can harness for a specific catalytic triad or catalytic activity that we want to introduce inside the pore. So now that we have this information about also the mechanism, as the question was saying, now we know which amino acids are essential for that. So it's more information to give it to Pele to get a better result for a specific ligand that we want to target. And also, now that we know which amino acids are exactly interacting with the membrane lipids, we can change those to change the lipid specificity of the pore. So now actinoporins in their natural state, they recognize sphingomyelin. But if we want to make them specific for another lipid, because we know that some cells implicated, for example, in cancer or some pathology, they have a specific lipid exposed, we can target that lipid with our modified nanoreactor. So this new structure is going to give us a whole new field to play with.
The next question is, what are the specific advantages and limitations of using nanodiscs compared to liposomes for studying pore-forming toxins like actinoporins?
Yeah, so we started to use nanodiscs first because you get a soluble structure even inside the membrane. And the advantages I already mentioned, like controlling the lipid composition, that in our case is really important, and also getting a more homogeneous environment. And actually, this is both a disadvantage and an advantage. Nanodiscs have a very restrained size. So for some things, it can be a disadvantage in the sense of you lose information about the dynamics of the process. But thanks to this size constraining, we got the intermediates of the pore formation, because our hypothesis is that in liposomes that we also used in this work to get the cryo-electron microscopy. In liposomes, you have enough space, and then the dynamics of the pore is so fast that you cannot see the intermediates, but we could see that into the nanodiscs. So at the same time, you have a size restraining that allows you to capture those intermediates, but you lose the dynamic processes of clustering. So actually in the work I didn't mention it, but we used both. We used nanodiscs and liposomes to get the resolution, the atomic resolution structures.
Did you ever use copolymers for this approach? Because then you would have the nanodisc, but also the flexibility.
Yes. So actually now, because I mean, we knew about the availability of the polymers, but we haven't tried them yet. But now, thanks to Cube, actually, we have a few samples to play with, because it would be great, as you said, to have a bigger nanodisc where I can control the lipid environment, and at the same time, I have enough space to capture all those dynamic processes. So yeah, we cannot wait to try the polymers for this.
Great. And the next question is, how do you add purified protein into empty nanodiscs? What is the advantage of this method over forming nanodiscs with your protein of interest in one reaction? I mean, this would, yeah.
Yeah, so we just add it to the proper ratio, a nanodisc of an actinoporin. We just add it, and then we just let them sit for half an hour or an hour, and the pore is just spontaneously formed. And I don't know if it's an advantage over forming the nanodisc in one reaction, but we wanted to emulate the biological process. So because I know that for people that work with real membrane proteins, I mean, actinoporins are membrane proteins, but they are also soluble, so they are quite special. But if you work with a membrane protein that is not soluble in aqueous solution, you have to put it in the initial mixture before detergent removal. But that's not what happens with actinoporins in nature. So we wanted to emulate the real process. That's why we decided to have the empty nanodisc and then to add the actinoporin, same as we do when we prepare liposomes, for example.
So far this was all questions, but maybe if some questions will still come up, we can hand them over to Sara per email and she would be still able to answer. Yeah, and do you have any suggestions or something you wanted to say still? Sara?
Suggestions, sorry?
Yeah, do you want to share any information still? Because so far we have no further questions.
Oh yeah, sure. So I shared with you with the company both articles, the one that I showed in the presentation in Nature Catalysis, and also the new one in bioarchives with the atomic resolution structure. So those are available online. So people, if they want to get further information, and you have my contact in case you think of a question afterwards. And yeah, with that, I just want to thank Cube Biotech again for this opportunity, and the audience for their attendance and their questions. So thank you.
We have one more question now. So the next question would be then, beta barrel pore forming toxins are more stable after forming the pore. Is there any advantage using alpha helical pores to design this catalyst, or you could also use the beta ones?
Yeah, very good question. Thank you, Alejandro. So basically, we used this protein because it was the one that we were working with. And Victor Guayar had the information of fragacetoxin C, and that's why we started there. We had no idea if the thing was going to work. So we just started there because it was easier for us. But now that we have that proof of concept, we are thinking of turning to beta pore-forming proteins. We are trying to get. We are working already into getting mutants of perfenolysine O, which is a pretty big pore forming protein, and we want to try that in order to get bigger pores that can handle bigger micro and nano plastic particles. And sure, we are very excited to explore that option.
And then thank you again for this really, really interesting research topic, and yeah, for the great work you did. Really impressive, your structure resolution. And yeah, then hope to hear from you again. And bye.
Thank you. Bye-bye.
Bye.